Evidence suggests that intervertebral disc degeneration, IVDD, can be induced by Propionibacterium acnes, P. acnes, although the underlying mechanisms are unclear. In this study, we analyzed the pathological changes in degenerated human intervertebral discs, IVDs, infected with P. acnes. Compared with P. acnes negative samples, P. acnes positive IVDs showed increased apoptosis of nucleus pulposus cells, NPCs, concomitant with severe IVDD. Then, a P. acnes inoculated IVD animal model was established, and severe IVDD was induced by P. acnes infection by promoting NPC apoptosis. The results suggested that P. acnes induced apoptosis of NPCs via the toll-like receptor 2, TLR2/Zhou N-terminal kinase, JNK, pathway and mitochondrial-mediated cell death. In addition, P. acnes was found to activate autophagy, which likely plays a role in apoptosis of NPCs. Overall, these findings further validated the involvement of P. acnes in the pathology of IVDD and provided evidence that P. acnes induced apoptosis of NPCs via the TLR2/JNK pathway is likely responsible for the pathology of IVDD. Introduction. Intervertebral disc degeneration, IVDD, produces a series of clinical symptoms, such as sciatica, low back pain and physical dysfunction, all of which drastically affect quality of life and work productivity of affected individuals and significantly increase the burden of medical treatment. 1. However, the etiology and pathophysiological mechanisms of IVDD are not well understood and emphasize the need to further investigate IVDD for better therapy. Traditionally, excessive mechanical loading, a nutritional disorder, traumatic injury or genetic predisposition is considered the main etiology for IVDD1. Recent studies have proposed bacteria-induced disc degeneration too because low-virulence anaerobic bacteria, such as P. acnes, were found to latently reside inside non-pyogenic IVD. In these patients, the prevalence of P. acnes in IVDD ranged from 13 to 44%, 3,4, 5, 6, 7. Further epidemiologic investigation and animal experiments suggested a link between bacterial infection and IVDD 8,9, 10, 11. Therefore, the theory of bacterial disc degeneration has drawn increasing attention. P. acnes, a microphilic or anaerobic gram-positive rod-shaped bacterium, is an important opportunistic pathogen that causes several diseases, such as endocarditis, prostate cancer, prosthetic joints, and orthopedic device-related infections and sarcoidosis. 12. Moreover, a growing body of evidence suggests that P. acnes is capable of growing and reproducing inside IVD. 11. In our previous study, P. acnes colonies were identified in non-pyogenic degenerated IVD by anaerobic culture and histological observation, and the prevalence rate of P. acnes in IVDD was 21.05%, 1676-13. Additionally, inoculation of P. acnes into normal rabbit IVDs induced severe disc degeneration and mitotic changes. 8. Therefore, P. acnes was thought to be a potential pathogenic factor for IVDD. However, the mechanisms by which P. acnes induce IVDD are unclear. Studies have suggested that cellular loss caused by excessive apoptosis of disc cells, especially the death of nucleus pulposus cells, NPCs, could play an important role in IVDD 14, 15, 16. NPCs are known to resist mechanical loading by synthesizing extracellular matrix, ECM, and thus maintaining the stability of IVD. Many complex and interdependent factors have been implicated in the excessive apoptosis of NPCs. 15. Thus far, studies examining the apoptotic signal transduction pathways of IVD cells have mainly focused on 3 apoptosis signaling pathways: the mitochondrial pathway, death receptor pathway, and endoplasmic reticulum, ER, pathway. 15. The mitochondrial pathway is activated by various cellular stresses and numerous apoptotic signals and is important for IVD cell apoptosis, which occurs during IVD degeneration 17. Because the etiology of P. acnes-induced IVDD is less well understood and the death of NPCs plays an important role in IVDD, here we investigated the potential relationship between P. acnes infection and NPC apoptosis. We also explored the specific signaling pathway responsible for the apoptosis of NPCs. To our knowledge, this is the first study to investigate the relationship between P. acnes infection and NPC apoptosis, and our findings provide new insights for the prevention and treatment of degenerative disc diseases.